Alright, this video is for star number 6, classification. The first thing for classification, we're looking at the 8 levels of classification. So remember, when we're looking at evolution, we're saying that all living organisms diverged from a common ancestor. So when we look at the 8 levels of classification, these are going to be based on actual DNA evidence. All of 7th period is staring at me as I record this. They're going to be based on DNA evidence or amino acid evidence or body evidence, like a comparative anatomy. So we're using different pieces of evidence to prove a common ancestor and to prove that almost all living things are going to be related in some way. So in order to prove that relationship, we group them into 8 levels. Remember, we've got Dear King Philip came over, oops, great soup. So writing that on your paper about Dear King Philip came over for great soup will help you to actually remember the order that it goes in. Please make sure you do know the order. That would be a kind of a silly question to get wrong in that STAR test. Okay, so let's look at the 8 levels. In this picture that you've got right here, you're missing the topmost level, which is domain. The reason why is domain is the most general level, and as we go down to species, it gets more specific. We usually leave off domain because it is so general. So on the picture, again, remember, as we go down, we go from general to specific. And the idea is that the more related you are, the more levels you will have in common. So if we look at the picture, you've got grizzly, black bear, panda, fox, squirrel, coral snake, and sea star. All of those are going to be in the same domain, in the same kingdom. Remember, we're missing the domain up here. But if we notice, a sea star is very different from the other ones. So as we go down to the next level of phylum, we get a little bit more specific, and I'm looking for just the ones that have a backbone, so the sea star does not, so we lose him in that level. That means everything that's in the same phylum is more closely related than, than the things that were in the same kingdom together. When we go down to the class, we're looking for just the mammals, so we lose the snake in that point. So we lose the snake at that point because he is not a mammal, so everybody who stayed together in the same class is even more related than everyone who's in the same phylum or everyone who is in the same kingdom. So as we keep going, we get more and more closely related. So as we get down to the genus, notice only 2 are left. That means these 2 are the most closely related out of the list of things I started with. When we get to species, which is the most specific, I only have 1 animal there. Why only 1? Because each species is so specific, it only describes 1 type of living thing. So in every species, you only see 1 example of an organism. You won't see multiple because it's so specific, it only talks about 1. So the more related you are, the more levels you'll make it through together. And the last part of this is scientific naming, which is called binomial nomenclature. For binomial nomenclature, every species has a 2-part name, and it's based on the 2 most specific levels. So the 2 most specific levels is the genus and the species. So the name of the grizzly bear, its scientific name is Ursus arctos. How did we get that? Ursus is the name of its genus, Arctos is the name of its species. We put those 2 words together, and there we have our binomial nomenclature for this grizzly bear. If I say Ursus arctos to anyone who is familiar with the scientific naming, then it's going to be the same name for all grizzly bears. No one will be confused. Why do we have to have scientific naming? That allows us to prevent confusion. Because remember, we look at a cougar. A lot of people have a different name for it, a mountain lion, a cougar, a puma, but its scientific name is only 1. So we avoid confusion in that way. Alrighty, so when we look at the domain and the kingdom, there are 3 domains we can classify all living things into, and then there are 3 domains and 6 kingdoms that we can classify all living things into. For the domains, our first one is going to be bacteria, archaea, and our last one is eukarya. For bacteria and archaea, these are going to be prokaryotic cells. Eukarya is going to be eukaryotic cells. There are a couple of things that are on this picture that are not included on yours, so as I go through those things, please make sure to add that to your graphic. Alrighty, so our 3 domains, we've already listed them. The next level, so domain kingdom is the next one. There are 6 kingdoms: eubacteria, archaea bacteria, protista, fungi, plantae, and animalia. Remember, if it has the word bacteria in it, then it's going to be prokaryotic. No organelles, no nucleus, anything like that. Alrighty, for eubacteria, let's go over it again. It is prokaryotic. It is unicellular, which means just 1 cell lives its life by itself. It doesn't collect together. They can be auto or heterotrophic. Remember, auto means it makes its own food. Hetero means it has to eat stuff. Does it have a cell wall? Remember, this is new on your picture. It has a cell wall made of something called peptidoglycan. Our next one is Archaeobacteria. Archaeobacteria is in domain Archaea. Again, it is prokaryotic. It lives in a very harsh environment. So the biggest difference between Archaea and you is one lives in a harsh environment, the other does not. It is unicellular, it can be auto or hetero, and it has a cell wall without peptidoglycan. And that's the second difference between eubacteria and Archaeobacteria. Alrighty, the next ones are going to be in domain eukarya, so we're looking at all eukaryotic cells here. Our first one, I'm going to skip protista for a second. Our first one is going to be fungi. Fungi, remember, these are our decomposers. There we go, took a while. So those are our decomposers. They break down dead matter, like dead plants, trees, things like that. They're going to be multicellular, so a bunch of these work together to form one mushroom. They are going to be heterotrophic. Again, they are decomposers. They are immobile, which means they cannot move anywhere. By the way, we're going to add that immobile. We're going to add that over here to Archaea. Sorry, I forgot that one on there. Alrighty, so going back to fungi, they are immobile, they do not move around, and they have a cell wall that's made out of something called chitin. Alrighty, our next one is going to be plantae. For plantae, again, eukaryotic, multicellular, autotrophs. They make their own food, they're immobile, and they have a cell wall made out of cellulose. Remember, if the word ends in OSE, it is going to be a sugar. Our last one for that we're going to talk about right now is going to be Animalia, eukaryotic, multicellular, heterotrophic. We have to eat stuff. We are mobile, and we have no cell wall. Now, our very last one, the one I skipped earlier, was Protista. Why did I skip Protista? Protista is the only one in eukarya that does not have specific characteristics assigned to it. Why? It's basically the catch-all, meaning if I don't know, if I found something, I discovered it, but I don't know if it's a fungus, a plant, or an animal, but it is eukaryotic, I just stick it into Protista. So notice they're all eukaryotic. That's the one thing they have in common. Some are uni, some are multi. Some are auto, some are hetero. Some have walls, some don't. So it's very diverse in this group because it's the catch-all for this for the catch-all kingdom. Alrighty, the last thing you have on there is a dichotomous key. Why do we use dichotomous keys? It's used to identify unknown organisms. Remember, you must follow it step by step. And so let's start with the animal that you have, or the organism you have in front of you. We're going to find out is what phylum it is a part of. So the picture that you have is a little bit different, but this is the exact same thing that we're looking at. Alrighty, so 1A says possesses segmentation. Segmentation means that its body is split up kind of like a worm. So it's got little lines and little parts to it. We can see, one second, we can see that it does not have any lines dividing its body. So we are not going to go to 2. We're going to look at 1B, which says lack segmentation, which is true. We're going to go to 3. So I skip 2A, skip 2B, I go down to 3A and 3B. 3A says it has a foot, a radula, an arm, or a shell. I don't see any of these things, so that is not correct. I look at 3B. It lacks all of the above, true, and it is dorso-ventrally flattened. Notice this organism is extremely flat, okay? So it is flattened. This is the name of the phylum. This is going to be phylum Platyhelminthes. Please make sure you're familiar with those words, dorso-ventrally flattened, radially divided, things like that. Make sure you understand those definitions, and that's going to be in your practice problem.